Naegleria fowleri, commonly known as the brain-eating amoeba, is a unicellular and free-living organism found in warm freshwater environment. It is thermophilic, meaning it thrives in high temperature. Although rare, it causes a severe brain infection called primary amoebic meningoencephalitis or PAM, which is almost always fatal. The organism has three life stages: trapezoid, which is the active form; flagellated form used for movement; and cyst, the dormant stage. This organism falls under the kingdom Protista within the phylum Percolozoa. It belongs to class Heterolobosi and order Schizopyrenida. The family is a Vulcanphiidae, the genus is Naegleria, and the species is Fowleri. This classification helps us understand its evolutionary lineage and biological traits. The scientific name Naegleria fowleri honors two researchers, Naegler, who studied amoeba with biflagellate stages, and Fowler, who first isolated it from human brain tissue in fetal cases in Australia between 1961 to 1965. It was officially named in 1970. The name also reflects its dual morphology, which are amoeboid and flagellated form, and its role as a deadly human parasite that causes PAM. And Fowleri has three morphological stages: trapezoid, cyst, and flagellated form. The trapezoid is the active and feeding stage, usually about 10 to 15 micrometers in size. It moves using lobopodia and contains granular cytoplasm with a visible nucleus and nucleolus. In harsh environment, it forms a cyst, which is 8 to 12 micrometers and has a smooth single-layer wall. This stage helps the amoeba survive adverse conditions. When placed in a distilled water, the trapezoid can transform into a flagellated form within minutes. This form is pear-shaped with two flagella and allows it to swim to a more favorable environment. In the environment, Fowleri feeds on bacteria and fungi through phagocytosis. Inside the human body, it becomes deadly by using its phagocytosis to nibble live brain cells. It enters via nasal cavity during water activities and releases cytolytic enzymes to digest host tissue, which leads to inflammation and brain damage. As I already mentioned in introduction, Fowleri is typically found in warm freshwater, and it can survive lakes, rivers, hot springs, and even swimming pools with poor disinfection. Its optimum growth is at temperature above 28 Celsius and can survive up to 55 Celsius. It also thrives in low turbidity and alkaline water with organic matter. Fowleri reproduces asexually by binary fission during its trapezoid stage, where the cell divides into two identical daughter cells. This stage is metabolically active, allowing rapid growth and division. When exposed to environmental stress like nutrient depletion or lower temperatures, it does not reproduce, but instead transforms into either a flagellated form or a cyst. The flagellated stage is for mobility, while the cyst stage enables dormancy and survival until condition becomes favorable again, at which point it returns to trapezoid stage to resume reproduction.
Moving on to survival, host, and invasion of Naegleria fowleri that is highly adaptable. It switches between forms like cyst, trapezoid, or flagellate, depending on the environment. Once inside the body, it uses surface proteins to avoid immune attacks. It also secretes enzymes called metalloproteinases that break down brain tissue. This invasion causes severe inflammation, leading to death within just a few days. Next, public health significance. This parasite causes primary amoebic meningoencephalitis, also known as PAM, which has a fatality rate of over 90-70%. Due to its extreme danger and rapid progression, the World Health Organization (WHO) classifies it as a priority B pathogen. It has been reported in more than 39 countries, making it a global concern. Now, I'm going to discuss about the significance of Naegleria fowleri in water treatment. Naegleria fowleri has been found in treated water when disinfection is inadequate. It can survive in pipes, water tanks, and even splash pads. To control it, the level of free chlorine must be maintained at or above 0.5 milligrams per liter. Regular monitoring and proper system maintenance are crucial to prevent contamination. Naegleria fowleri is a valuable model for studying brain-invading parasites. It helps us understand how pathogens evade the immune system. It is also driving research into faster diagnostic tools and effective treatments. Overall, it plays an important role in both medical science and public health studies. For detection and sampling methods of Naegleria fowleri in the environment, scientists often use PCR testing. Samples are collected from water, soil, and places like sink traps. They are mixed with amoeba saline, which helps maintain osmotic balance and keeps the organism alive for testing. In the lab, the organism is cultured on non-nutrient agar and observed using light microscopy. It is stored at temperatures between 37 to 44 degrees Celsius to mimic the human body. Symptoms of infection include fever, headache, nausea, stiff neck, hallucinations, and eventually coma. The disease progresses very quickly, so early detection is absolutely critical. In conclusion, Naegleria fowleri is a rare but deadly amoeba that poses a serious threat to human health. Its presence highlights the urgent need for increased awareness and the implementation of preventive water safety measures. Given its fatal nature and limited treatment options, it also underscores the importance of ongoing research and investment in public health to better understand, detect, and manage potential outbreaks. Here are some references that we used throughout our research.